Well, good morning, Sheila.
Good morning, Joe.
It's my pleasure this morning to introduce and interview Dr. Sheila Muldoon, who's been a longtime member of professional societies in anesthesiology and associated with the Malignant Hyperthermia Association of the United States. Sheila's had not only a distinguished career, but also a very productive one scientifically, clinically, and from an educational perspective. So Sheila, first of all, thanks for taking the time to have me interview you today.
Pleasure, Joe.
Good. Let's start. We know not as much about your background as we'd like. So I know that you originally received your medical training in Ireland.
Yes.
Why don't we start there?
Yes. I obtained my medical degree from University College Dublin in Ireland. And after that was an intern, did my internship there and some time in obstetrics, which I was thinking of doing. I didn't like it as much as I thought, so I joined an anesthesiology training program at one of the teaching hospitals in Dublin called the Mater Hospital. And I did that for a year, and I liked it very much, and would have continued to do that, except I got an offer of marriage from my boyfriend. And I decided he wanted to go to train in America, and he had got a position as a fellow at Mayo Clinic in Rochester, Minnesota. I had no idea what I was getting into, but I said, well, I'll apply for a job. I'm sure they won't take me, but they did. And I ended up in Rochester, Minnesota in October 1966.
So where did you find the weather was harsher, in Dublin or in Rochester?
Shall I say, the first winter I was there was snow in October. There was 3 weeks of below 30 to minus 30 degrees in January, and there was snow in May. I think that probably speaks for itself.
I think so. Well, so you first came to the States and you were at the Mayo Clinic.
Yes.
And then eventually you were recruited to the Uniformed Services University. Tell me a little bit about that.
Well, can I just back up?
Sure.
After I finished my training in anesthesiology, because I had done a year in Ireland, they said that I only needed to do 2 years of training. But they did offer residents who wanted to get extra training in research an opportunity to study for a degree in physiology and anesthesiology.
Mm-hmm.
I signed up for that. And it was during that year where I got an opportunity to go to the lab, learn some techniques, and also to learn more physiology.
So let's talk about that experience for a minute. In going into the lab and learning about physiology, did it specifically start right in muscular physiology or was it general physiology?
Well, the course was general physiology, but the lab I went to work with was run by a man called Dr. Richard Thai, who was extremely well known. And at that time, his main interest in life was, or his research interest, was in working out the oxygen consumption of different organs, whole body oxygen consumption, and the effect of anesthetics on it, and then looking at specific organs like the brain, muscle, and kidneys, whatever. So by the time I came along, muscle hadn't been looked at. So my boss said, we want somebody to look at skeletal muscle, so we're going to do these whole animal experiments. And there was, of course, a very well-trained technologist to dissect the muscle, so we perfused them with anesthetic agents and looked at oxygen consumption, whole body and muscle oxygen consumption. And for that, I got a master's thesis. Now, the master's thesis, Mayo Clinic at that time didn't have a medical school. So my degree, a master's, come from the University of Minnesota, which had an affiliation with Mayo Foundation. And then I was recruited to the staff and stayed there for 7 years. Mm-hmm. And during that period on staff, it was hard to get funding to do research because I was hired as a clinician, but I kept plodding away and had very good experiences working in the Department of Physiology with Dr. Shepherd. And eventually I got a small grant by the Minnesota Heart Association, and that gave me a day a week. And so then with a little bit more hard work and some luck, I got that extended, and eventually I got an NIH grant.
Great.
And this was all in the area of the control of the vasculature in humans by anesthetics, and how did it affect norepinephrine release and metabolism. So one of the things I learned in that years was in Dr. Shepherd's lab was how to dissect blood vessels put them in an isolated organ chamber, and study their responses to drugs, including the muscle tension.
Mm-hmm.
And that was what we were funded to do. And that's that brought that grant with me to Uniform Services when my husband wanted to come to Washington. And I could also bring the equipment because it was purchased on a grant. But I was determined to continue with my vascular smooth muscle until one day we got a new chair and in anesthesiology, and that was Colonel Robert Watson, and he had a completely different set of problems. They were very supportive of my work and delighted to have me, but they said, just switching to Dr. Watson, he was chief of anesthesiology and operative service at Walter Reed.
Mm-hmm.
He was also consultant to the Army Surgeon General. Now this was, and he had to write reports about not just what was happening at Walter Reed, but around the United States, and send reports about the surgeon to the Surgeon General. And Bob, who I'll call Dr. Watson from here on in, he started telling me about these cases that were out in small army hospitals in Wyoming or Montana or anywhere, and they'd have a malignant hyperthermic episode. We're now talking 1980.
Mm-hmm.
There was no dantrolene available at the time, or it had just become available, but there was no M-House, there was no hotline. So the person they called would be Dr. Watson, and he in turn would call Beverly Britt, because she was the acknowledged expert, and she was tremendously helpful. But as Bob said, you know, she's getting rather tired of these calls and thinks I should be doing something about it down here ourselves. And so he said, I wonder, could you look into it? And I said, I think it's a very complicated disease. I don't know anything about it, but I'll read up on it. And I read a little bit and I thought, my goodness. But I had one bright idea and thought, I knew that Children's Hospital had been part of one of the centres that had been in the FDA program to approve dantrolene.
Mm-hmm.
So I thought, I'll call up and see, can I go visit them? And perhaps they'd give me some pointers. What do they have patients that I could talk to? And so I did that, and I went and saw them. And Dr. Epstein was the chair, and I told him what my mission had been and what the request of Dr. Watson was, and I said, I thought, I guess maybe you could help me. And he opened a drawer and he took out a stack of papers and he showed me some results from tests that he got from another university where he had entered into a contract to test patients. And it was Columbia.
Mm-hmm.
And they were going to doing single muscle fibers on samples he'd send, and also measuring cyclic AMP. The problem for the clinician like Dr. Watson and another chap, Matt Becker, was that these were not easy to interpret. So he handed them to me and he said, can you interpret that? And I said, no, Bob, that's not, no, I can't interpret that. That's not the test I have in mind. I want to look at whole muscle. That's the way it's going with Gronert and other people and Britt.
And that was your expertise. You'd already learned to dissect larger pieces of tissue. Mm-hmm.
Yeah, right. So as soon as I said that, I still tell everybody this, Dr. Epstein said to me, are you really telling me you can do this? And I said, yes, if you doubt it, call Jerry Gronert.
Mm-hmm.
I hadn't worked with Jerry, but we knew each other pretty well. And he said, well, I have somebody I want you to meet. And he introduced me to a lady called Dr. Gloria Eng, who was a neurologist and who was the head of physical medicine. And she had been interested in malignant hyperthermia because many of the children who had died in the pre-dantrolene days there were children who had myopathies, who Gloria had been managing their rehabilitation program. So she was a tremendous resource.
So here you have a stipulation from your chief to think about and provide some new service for these orphan military individuals that are at different outposts.
Right.
And in your own backyard at Children's in Washington, you happen to have not only interested but experienced people with malignant hyperthermia whether it was a single disease or a syndrome.
Right. No, it was, they were encouraging, the Children's Hospital, and we formed, and helpful. They had patients I could see, and they wanted me to test for them. So we got going, and we formed a partnership, which is still going to this day. And it's, I'll come back to it when we're talking about their contribution. I can't speak enough good about them. And also Matt Becker, who was later a hotline consultant and worked with Henry and the group of us.
Now, how many years after you started this before the issue was before the Department of Defense about the eligibility of patients to enter the military or medically be discharged from the military because of potential heat issues?
Oh, it was already there.
Uh-huh.
I actually tried to find out when the DoD regulation had been put in, and I asked a historian. It was before the issue of Uniformed Services University being opened, probably in the late 70s. I think Dr. Rosenberg has referred to the international conferences that had been held, workshops that had been held. That had got the word out that this was a disorder that could be lethal and needed immediate treatment. So that had improved people's education a lot.
So I'm going to ask you, in this interview, if you were talking to non-medically trained people, let's talk about what might happen in battlefield medicine that would predispose these soldiers to trigger a malignant hyperthermia episode.
Well, let me just back up one point.
Sure.
I mean, it's very unfortunate because if you are recruited to active duty, you may have a malignant hyperthermia trait and not know it. As you know, if you don't have a family history, or you haven't had a death in the family, or haven't had parents who have had surgery that got these anesthetics, you could honestly say, we don't have it. If you do have it, and you get on active duty, and you have an episode, you are going to be disqualified. Because, like you said, taking care of patients in the battlefield theatre would just put others at risk. And yourself, you're also at more risk because suddenly the demands to keep your job, which is to keep a high level of fitness, and often you're going to be working in really hot environments, like we have.
Sure.
And so you're at additional risk.
So let's go into the early to mid 80s or even the late 70s for a second, and we talk about battlefield medicine, and a soldier is injured. He's about to have emergency surgery. The state of the art at that time would have included what drugs to, quote, anesthetize him?
Oh, the inhalation anesthetics were the main drugs. And because, often if you're in a battlefield condition, they may have had to bring in small vaporizers. But even if you were in a major hospital, inhalation agents, pretty much the same drugs that we were using in civilian life. And succinylcholine was widely used.
Sure. So just like trauma victims here in the United States, if we need to establish the airway as quickly as possible, the drug of choice was succinylcholine.
Absolutely.
Yeah. And so, to the average soldier, they might not think that they're at any increased risk for medical complications, but someone with MH susceptibility is going to be given the, quote, full trigger as a part of the standard of care because that's the safest way to actually take care of them.
Yes, that's right. And also, as dantrolene became approved, then people had to stock it and bring it with them. Of course, that was an extra burden for people because, as you know, we needed 36 vials, and we also needed to bring sterile water, and we had to have, train people up how to use it, and that was another burden. But the real challenge was what to do with a critically injured soldier who may develop a fulminant malignant hyperthermia when you have maybe a whole casualty full of seriously and high-risk soldiers.
Sure. Now take me through these first few years to the first many years at USUS and tell me about the development of the MH testing lab. And was that conceived because the military wanted to be able to do it themselves, or was it really your initiative?
It was my initiative, because while Dr. Watson was talking about getting some help for, these people who were out on small bases and treating them, and I said to him, one of the things, Bob, we have to find them, because it is no point in them being anaesthetized out in a remote area if they don't have the drugs to treat it. At that time, there hadn't been a directive sent to stock dantrolene everywhere. I'll come to that in a minute. But so I said, we have to find them. And he said, you're absolutely right. You have to find them, and then we'll improve the treatment. In the meantime, the same person, Dr. Bob Watson, he had followed the progression of dantrolene. And he realized that the med centers in the military would stock dantrolene. Again, the concern was the smaller hospitals. So he was able to write a directive through the Surgeon General, the Army Surgeon General, which was approved almost immediately. And that directive was picked up by the Surgeon General of the Navy and Air Force, and absolutely described that anywhere general anesthesia was to be used, they were to stock 1 complete dose for an adult of dantrolene.
So now
and that's 81. So we were
81. Okay.
Bob was well ahead of the curve of, as soon as it was approved, and we had this conversation.
So here you are as a young assistant professor, and you've been charged with find them. What strategy were you thinking of employing to find these susceptible individuals in the military?
Well, we gave talks, and of course there was lots of patients that would call Bob or the consultants. But we set up a department structure. The medical school was very new. We weren't even in our own department space until 1980. So I had first been in the Department of Physiology and working in anesthesiology. So then as soon as we got a telephone number, we got some flyers posted and started advertising or telling people about it. And the services got the word very quickly.
And then did the commands refer in patients with heat stroke or heat exhaustion?
No. What the command sent us was patients who had mostly had malignant hyperthermia, either had survived or their relatives, their dependents who were entitled to care would be referred for testing.
Uh-huh.
And sometimes they would send me a whole family. That happened once. They sent members of family from overseas. You can imagine what a price.
I've seen some of those family pedigrees that you've shared with us in the past, and they can be quite extensive.
We quickly it took a few years to get everybody on board. We want to see the index case, if they have survived, or their first-degree relatives, and then we will be able to tell you. But our main result was to try and get them, and I began to serve on the hotline, which I think I began in 82, I started. That was a great way also to spread the word, and got me a lot of contact with people who called in, and the word got out.
So then, in addition to these kind of organized directives that you have to respond to, and volunteering your time with the hotline, you began a rather prolific scientific and publishing career. I want to tell us a little bit about some of those academic details.
Well, I don't know whether it's that prolific.
I think it is.
Well, first to go back to the military. At the training programs in that period, in the 1980s, you could get 6 months of research as part of your training. And people like Watson and other chiefs at the Navy and Air Force said, look, we want to learn more about this, so we're going to send somebody to work in your lab. And they were usually young anesthesiologists. Some had some scientific training, most had none. But we were able to hire, on our budget that we got from the university, we was able to hire a chemist, because I knew I would want to monitor the halothane concentration in the bath, of which, when I got these whole muscle bundles that I could test them, I would want to know what the halothane concentration, just as I needed to know when I was doing my vascular rings. And I also hired a good young person to work in biology. And then one day I got a great postdoc, and that was Dr. Doyster. And she came to work for me. She was interested in muscle, and she also did studies in the pig, which we did. And so publications came from having talented people work and doing some, and still had our grant, our NIH grant. But we got some money from the university. But the DoD never put much money in. What they did was supported my people, supported the staff, and we could buy equipment on a yearly basis. So it's now much bigger, of course, but that's what the basis was. So what I had to do was manage a budget and try and think ahead what we needed and do that. And things grew.
The answer to my next question need not be exact, but give me an idea about how many fellows or anesthesiologists you've actually trained over your career.
Well, the ones who came to do sick before the rules for training in anesthesiology changed, I'd usually have 1 or 2 a year.
Mm-hmm.
So probably go about 10 to 15. But then the rules about residency changed, and that year went to specialty training. Then we didn't have volunteers for the research. But out of that group, I got great people,
Mm-hmm.
and including one chap who worked on trying to get dantrolene into solution, and other guys which contributed to the program, and also helped with the hotline.
Mm-hmm.
Became hotline consultants,
Mm-hmm.
2 of them for a while.
Now, I don't want to move forward too quickly because I may be missing something that you want to cover. So please interrupt me and do that as a prelude if you need. But at some point, even before I was involved with MH, there was this specter of an idea about the genetics or the human genetics are beginning to improve enough, and we have some techniques. And you felt the opportunity to develop a genetic research opportunity with one of your collaborators, and we applied that together in a number of publications that we've seen.
Right, yeah. No, that didn't the gene for the ryanodine receptor gene was announced, I think, in 1990.
Mm-hmm.
So we had been working about 10 years and had a good cadre of subjects now, positives and negatives, and had got a handle on that. I went to a neurology meeting in Munich, I think shortly after the gene was announced, and I got the idea, look, we have to start doing genetics. Now, I was entitled to a sabbatical because I'd been more than 10 years working at my university. So I applied for a sabbatical, and I went to Würzburg. There was a talk at this thing, a geneticist, a professional geneticist, and a clinician. 2 guys gave a back-to-back talk, and I thought that's the way we should do it. And so I went to Würzburg, and I worked with Dr. Mueller and the clinicians. And then I came back. We already had the testing center going for the diagnostic thing, and set about, got the lab, one area of the lab modified for genetics, and then set about trying to hire geneticists. And the first one was Dr. Say. And he came from NIH, and he was brilliant. He was an MD, PhD, worked on the AIDS program. He was very interested in the immunologic aspects, and he knew how to work with B cells. So he thought, look, we're working on the genetics, but we need a less invasive phenotype.
Mm-hmm.
You have a good phenotype. We have patients who have been diagnosed with malignant hyperthermia, so we could use gamma and get B cells from them and see would they express the same gene, the ryanodine receptor, and they did.
Mm-hmm.
And he announced that at a European meeting, and it was a stunning discovery because he was able to, he analyzed the protein now because much less protein inside the B cell. Unfortunately for the B cell story, B cells are not muscle cells.
Mm-hmm.
They don't have a membrane that's, they don't do EC coupling.
Right.
And for MH, I think it's a muscle story. But many people use the B cells for testing and have used it. I think it's more used now outside of the United States. The Australians and New Zealanders have done a lot of studies, and we're probably going to go back to it.
Mm-hmm.
But it didn't catch on as a diagnostic test. It's still a good research tool.
Mm-hmm.
Dr. Say thought it would be both. He told me one day, Sheila, we have both the phenotype and genotype in our hands. Mm-hmm. Not quite.
It's a great hypothesis that just didn't come to fruition,
pan out,
right? Yeah. So tell me a little bit further, because we happen to know a mutual colleague, Kishka Sembuken.
Right.
And tell me a little bit about how you got started with her.
Well, she was actually a protege of Dr. Nelson.
Mm-hmm.
Dr. Nelson had a friend, another friend of his era from his, professional career, And that friend was looking for a job for a young geneticist who had to leave NIH having completed work there. She was a Mongolian. And Tom and this friend, who I can't remember, they were very impressed with this lady. And so Tom got her interested in starting to work on the genetics. And thank goodness he did, because a famous publication came of that. There was a malignant hyperthermic episode in a child down in North Carolina who was playing football. It was a 12-year-old boy, as I remember. And this child had had an MH episode about 6 months before. He'd been treated with dantrolene, made a good recovery. And then he went to play football on a warm day. And he got sick, and he asked his father to come off the field. When his father took him to the shower, he was so hot he could hardly touch him. And he couldn't speak because he had rigidity in his masseter muscles. So they tried to keep him going until he got to a hospital, but he died. You and Dr. Sambugan was working down in a clinic in another state, and I guess Tom knew about her work, and he was able, and a fellow called who might be sitting close to me, Dr. Tobin, published a paper with Dr. Sambugan and Dr. Nelson reporting the first MH mutation, which was a causative mutation, in a patient who was known malignant hyperthermia susceptible, who had an episode while playing football in the heat.
And so, you know, we were on the back end of receiving this patient because he was being brought to our medical center for his post-mortem exam. And that's when we got a call, and the pathologist asked us, how do you diagnose malignant hyperthermia? And the pathologists weren't aware, and we said there was no postmortem test, but we have this creative geneticist who might be able to do something with some genetic material.
Yeah.
And we did indeed find a causative mutation, and that did result in a publication, which I think opened a lot of people's eyes to say spontaneous MH really does exist, or exercise-induced does exist.
It opened up the whole field, Joe, because they put it in JAMA, which was read by a lot of physicians. And it just, and of course it was the first advent of genetics in the diagnostic field, and useful for patients who had died and for the family. The father of that child who died, he had not had an MH episode, but he had a history of muscle cramps and exercise intolerance. And but his brother, who came to us, the brother and uncle of the child, did not. So there were family members who were positive. You know, it was like the autosomal story that Henry was talking about. You know, one brother had the trait and passed it to his son. The other one did not. So, but it was a big breakthrough.
Well, then, not long after that, over the next 5 to 10 years, your continued collaboration with Kishka resulted in sequencing a very large percentage, or the entire ryanodine receptor gene, and screening North America for many of the causative mutations?
Well, after we got the lab set up and Dr. Say was working, I decided that even though we were getting a pretty steady number of patients to biopsy and do the caffeine halothane contracture test, I knew I wasn't going to have enough patients who would be positive. So I formed a multi-center study. And in that I had, not Joe at the time, but Henry, Dr. Rosenberg sent me samples. Got samples from Dr. Bronert when he was in California, and I think in Northwestern. So we were able to collect about over 100 patients who had had CHCT testing and who were positive. And we set out to do the genetics, I think, in about 1997. Because, of course, now I was changing gears. First I had a diagnostic test for which I did not need institutional review. But now I was going to do research. And we were setting up a research lab, not a CLIA-approved lab for genetics. And so that had to go through the IRB approval process at our institution, which is formidable. And they were very skeptical that an anesthesia department should be setting up this in the first place. And so there was lots of back and forth. But we got going, and Dr. Say and Dr. Sambugan then was between jobs, and she came back and stayed for a while. And she and Say did a bunch of patients. Then we published the first paper on those patients in, I think, 2001. It took a couple of years. But we were only looking at the known mutations. We weren't able to look at the whole ryanodine receptor or even the hotspots. So we were looking at checking for and we found about 20% or 22% or something like this of the patients who were CHCT positive. And this was a very limited panel. And then I began to think, we knew about these families who congregated in places like Wisconsin and Quebec. Then I looked at the population of where did most of my patients come from, and they came from the eastern, part of the United States. And of course, you look there, who were the main immigrants there? There were the Germans and the British Isles people. Mm-hmm. Yeah, and so we were on a good track for looking at, working with the European and watching their progress, because they were certainly way ahead, had many labs, mm-hmm. Testing and doing genetics, and working out and finding, they found the second gene, and Monnier did, and after McCarthy and after the ryanodine receptor.
Let me switch gears for a moment. In parallel with your academic prowess, you also became a staunch supporter and a member of the Malignant Hyperthermia Association. In this nearly 4 decades of its existence, and we're going to be coming up in just a couple of years to our 40th anniversary, tell me what you thought of the organization when it first started, and what has it become, and what do you see for it for the future?
Well, let me go back.
Sure.
When we were setting up the contracture test, even though I had experience with vascular smooth muscle, getting cut skeletal muscle fibers to work was not easy. We bought some pigs with the trait. We could see there were differences, but we weren't getting very consistent results in measuring tension responses to the drugs. And so I called Dr. Henry Rosenberg. He had been working in the lab, and he had trained in Bianchi's lab. And he came down and gave us some pointers. That greatly improved our technique. So from Henry, I think it was, I heard about malignant hyperthermia and setting up he was at that time looking for people or talking about setting up a helpline, which would have fitted right into what Dr. Watson was talking about years before. And I think I joined the hotline group in 1982, because I finished my 20 years in 2002.
Okay.
So back to Dr. Henry Rosenberg and his help, which we've always acknowledged and are grateful for. But he had given us very practical help, and that got us over a hump and introduced me to the serving on the hotline, mm-hmm. which was incredibly challenging in the early days, but so worthwhile. Mm-hmm.
So then, tell me about, after hotline exposure, the further introduction to the Professional Advisory Council or to the board, and other things you saw that matured over those couple of decades for MHouse.
Well, the registry, of course, at this time, let me just switch to the registry, was in Hershey and under very capable hands of Doctors Larrick, Marilyn, her husband. Then they left there, and Marilyn was just a prodigious worker. And she had collected a huge amount of data from everywhere where a malignant hyperthermic episode. She had a form called the AMRA, the Adverse Metabolic Response to Anesthesia. And she had really good help. But they left, and they were to start again in Baltimore, and they were promised a center that would take over their work. That didn't work out, but they got jobs there. But then that left the registry in a bit of a bind, mm-hmm. because they didn't have anybody to supervise the administrative staff there. So I think Henry asked me, would I go up there 1 day a week or every 2 days a week while they were looking for a place. It quickly became apparent that Baltimore wasn't going to work, yet the Larricks were working there, and so they didn't want to move. So I did that for a while. And that led to the board asking me, would I be temporary head of the registry? That was, I think, in 95. Mm-hmm. I think the temporary went on a bit too long. until 15 years or something. Yeah.
And that's when it relocated to Pittsburgh?
No,
Or when?
it moved to Pittsburgh, I think it moved to Pittsburgh about probably 2000. Yes. I mean, others might correct me on that.
But it was a lengthy temporary assignment.
But it gave me, I knew it couldn't stay there because there was a great upheaval going on in mergers and things between hospitals. Nobody knew in that area of Pennsylvania who they were going to be working with. It was a very troubled period with hospitals and who was going. And also the chair of anesthesiology, who had been very supportive, was about to retire. Mm-hmm. So that, we knew it had to move, and Dr. Rosenberg and I went out and looked for a centre. We looked at a couple of centres, but he eventually decided on Pittsburgh and Dr. Brandon, and they were very supportive of that. Yeah.
Great. And then, recently, I became involved in the relocation of the registry now going to its newest location, and I was actually curious to hear about the process of when you did this once before. We had a nice organized process most recently. So I'm curious to see how ad hoc was it back then, or were you just flying by the seat of your pants and you and Henry just had to wink at each other and say this is it, or did the board actually instruct you how to go about doing this?
The board were very involved.
Okay.
The board was involved. The registry was well established, and we knew it was an asset, and we knew we had to find the best place. We interviewed, two places. We went where Dr. Sambugan was working, which was a neurologic institute down in Arizona. Right, and they would have liked us, but there wasn't enough support there. We loved what she was doing, but there wasn't enough administrative support. But we really liked Pittsburgh, and the chair was supportive, the dean was supportive, but we were following the instructions of the board.
Good. And then when did you join the board?
About 1996, around that time.
And how would you say you've seen us grow over these last 20 years as an organization? Either
phenomenal.
Membership, scientific understanding,
Everything.
and support?
Oh, well, the membership has grown. Of course, the offices moved to the new office. I mean, they were in different places. They've always been... There was a change in structure sometime along the line when... and I can't remember exactly what year they moved to their present location. But no, the membership. But the real advance has been in the treatment and identification of malignant hyperthermia patients, and the commitment that the society has made, and recruitment of new hotline consultants, the whole process, recording them, making the material available. It's been phenomenal success.
If you had a crystal ball and you said, I can look forward 5 years or 10 years, what do you see as the next major accomplishment of the organization?
Well, getting the word out, we have to step back and say that there are still many physicians and nurse providers and managers, all in the scheme of things, both in civilian and military life, who don't know enough about malignant hyperthermia. The military are actually better off because, we teach it, it's hammered into them, it's a very relevant disease, and so they make sure that they know about it. The civilian sector, the doctors, I think, many anesthesiologists of my age, or maybe younger even, now are not faced with the same crisis that we had in the 1980s, where everybody feared it. So they've now taken it, I think, a little bit more casually, and so that's why deaths occur. So there is a big job still to be done in education. In the research area, I think we will have a non-invasive test in the next decade. Of course, we thought that.
Sure. We've had false starts before.
Well, we didn't understand the complexity of the gene.
Sure.
And as Dr. Rosenberg said in his talk, the large signs of the gene, and also the incredible difference of difficulty of understanding why a patient can be anesthetized 5 or 6 times with the same triggering agents, not develop any of the signs of malignant hyperthermia, and then take him in to do a hernia repair, perfectly fit person, and the next thing he's dead. That is extraordinarily difficult for the public. And when you and I talk to them about, the different aspects of the gene expression, and that, I don't think they get it. They do get it. Penetrance is now being better understood, and diversity. So there's an awful lot to be learned about the whole syndrome of malignant hyperthermia.
So that was a great segue into my next question. From your perspective as a scientist and as a clinician and as a professional who's been involved with this organization as long as you have, do you think we're treating or examining one disease, or do you think we're examining a syndrome of diseases?
A syndrome, yeah, a syndrome. Yes, a good part of the, maybe 70%, maybe not quite that high, of the patients who develop malignant hyperthermia in the operating room have a ryanodine receptor gene, whether it's been characterized or not, which is another challenge coming up to us. But there's a whole lot of people who don't have that. I have had patients who had full-blown episodes of MH whose anesthesia record I would show to anybody and they'd say that met the criteria, end-tidal CO2, up metabolite rigidity there, and we don't find a gene. We don't find a mutation in the major candidate gene, either of them. And the same is true in Toronto and the same is true in Europe. So we're missing something.
Well, and I want to bring up another field of yours that has been a strong priority. When I was first taught about MH as an anesthesia resident, we were taught that if someone successfully survived an MH crisis, there were no long-term sequelae. You were one of the earliest pioneer voices that said, we have people that have pre-existing muscle conditions with cramps or related symptoms, and these people that develop prolonged or sometimes intermittent but frequent episodes following an MH crisis, and they have a lifelong problem with symptoms.
Yes. No, I'll just back up. The first real insight I got into this was when two kids. We had a child of an Air Force active duty member, and this little boy had a malignant hyperthermia episode. Somewhere he was treated, well treated, but then he continued to develop awake episodes. Tremendous tension in his muscles, severe pain, high temperature, and mother would race to a hospital, and if he didn't get intravenous dantrolene, he was just about to expire. He was the father, when this happened the first time, the father was assigned to a base in Britain. So he was taken to Oxford and then to hospital in London, and they thought he had a neurologic disease. So then they did the rounds of neurology, getting, every good centre where there was pediatric neurologists, and there was no diagnosis. One man said actually that he thought he might have been a King-Denborough syndrome. Now in the meantime, the mother was just really living on the edge. When would this kid get another episode? Would she be able to get him in time? So one of the things is, so Marilyn Larrick actually picked up the hotline on one of these crises that the kid was having when they were in Texas. And she asked the doctor that she was dealing with, have you guys tested him for malignant hyperthermia? And they said no. And then she said, I suggest that you take him to uniform services. So the father brought him to Walter Reed. I got the Children's Hospital involved to also evaluate him, and they did an EMG on him to see and it was normal. His development in everything, he was just absolutely perfect little boy, that was running around 1 minute and the next would be just in severe pain and a rapidly rising temperature. But he was 5 years old when I did his biopsy, and he gave me one of the most positive small contracture results I've ever seen, over 9 or 10 grams in this small child. So then the mom said, I noticed that, because she'd been questioned by everybody, what is the prodrome? What do you notice before? She says every time he gets a cold, then he seems to get these muscle spasms. So she asked if we could put him on antibiotics, or could we put him on dantrolene to be prevented. So we took him into hospital. We got a compound chemist to make the dantrolene orally, and we sent him home. And the episodes decreased. They didn't stop. And the father was then assigned in the Midwest, and it was a very hot summer. And I don't know whether he had, it was a temporary measure. Obviously, we thought that he might grow out of it, but he died of being taken to the hospital on a very hot day with what looked like a typical MH episode. So, that case haunted me forever. And I always thought, if we don't solve this, we're going to have another one. And we did. And it was a different scenario, but we had 2 children then with... the other child didn't have an MH episode, but was having awake-like symptoms. And after several years working, Dr. Sambugan discovered that both children had the same ryanodine receptor mutation.
I see. Now, when the mom of the just slightly earlier child you were speaking of said that the prodrome was a URI or a cold,
that's what she thought.
Did that make anybody in the MH community suspicious that this might be an immune-related disease?
They did. They thought about it, but they didn't come up with any. Say when he came to work with me, and he was still working with us, said, "Look, I told you," but we didn't have any good markers.
And the whole science of immunology has advanced just as rapidly as genetics has. So there would be molecules now that we didn't even think we knew of back then. So I can understand that the limitation of the ability to work it out.
Right. No, that was my, it was tragic. And actually, the mutation that Dr. Sambugan found was characterized by the pharmacologist that's here in this city, whose name I'm blanking on right now. Bob, yes. Yeah. Right. And that led to a whole lot of interest in awake syndrome. But to go back to the less dramatic, but really tremendously painful and troubling that I see with adults, particularly young adults, who will be able to run, do leap tall, do anything the services want. And then they either have an MH episode or they might have a severe rhabdomyolysis of unknown etiology, and they don't get better. They don't get better. We can understand it, I think, in terms of the pathophysiology of malignant hyperthermia, because we know they have a leaky channel, and they're probably leaking calcium all the time and putting a stress on their, ATP. The ones with the rhabdomyolysis, and we didn't know, and that stimulated my interest in working with neurology and with sports medicine, and to the present grant that we have. And yes, heat and exercise can trigger an MH event. It's not that common. More common are the sequelae that Barbara Brandon wrote so well about, of how many people have muscle pain that goes. It's understandable if you have muscle pain and dysfunction after an acute insult to muscle, like your hotline, like your MH event, but for it to go on for months or years, and we really don't have any good treatment except oral dantramine. Excuse me.
Sure. With a nearly 4-decade career now in MH involvement and your scientific work, I think I might have to consider proposing to the board that we would make you one of the pioneering international ambassadors along with Dr. Rosenberg, and we're going to have to send you both around the world to keep up the message.
Oh, I think you do much better with Dr. Rosenberg.
I don't think he can visit every place himself, so I think we're going to need more than one.
No, we've been on different panels, and and we get calls, and I think the number of calls. Well, Henry had a very active centre until he went to his new job. Excuse me. But we get a lot of calls about about patients about MH and about the exercise-related syndromes, and now have, as I say, a collaboration with sports medicine and have a protocol to deal with those, which is pretty much like the one in Toronto. We don't biopsy patients with exercise-related syndromes just because they haven't had an episode in the OR, and so there's not a nice anesthesia record to tick off the checks and see that they met all the things. But we have a group of people who are sports medicine and neurology, and they evaluate them. And we don't do biopsies do CHCT on those people unless there's agreement that there is no other potential diagnosis that could be reached.
So what's your recommendation for a family where there might be more than one member who have experienced repeated episodes of exercise-induced rhabdomyolysis? would you refer them only to a neurologist first for a workup?
Yeah.
Or would you just accept them based upon the percentage that we know will be CHCT positive and just biopsy them?
No, I would send them and do send them. And I have always sent them to neurologists or heat specialists, but mostly neurology. And sometimes you do find that they have a myopathic condition. And we've found CPT deficiency in patients walking around, and that can explain their intolerance to exercise and heat. But not many. And
what's your recommendation for a family where they have persistent hyper-CKemia? Say it's substantial. Say it's 1,000 to 2,000. It's not it's not just 301,
Right.
but it's up there and it's persistent.
yeah, well, I used the data that Mayo Clinic had, where Margaret Walensky
Yep.
actually looked at their cadre or their cohort of MH patients, their positives, and about 50% of them had an elevated CK. I haven't done that same study with the military, because I think that was an excellent study. So yes, we're going to find people with hyper-CKemia, but most hyper-CKemia are not RYR1 positive. If you have family history and you have an elevated CK and you have an MH event, you have the diagnosis.
You got the whole
But
shebang.
unfortunately, those people don't seem to call my office.
Yeah. Well, I think that somebody has said the diagnosis has been put to rest and they never call us for advice about what to do because the story seems clear to them.
Right, and and there's multiple causes for hypercemia, and plus, the advent then of the Lipitor drugs have increased that group, which is also a group of interest. But I'm not sure there's a good association, but I don't know that it's more than that.
Sure.
Although there are people who are working on it,
Sure.
but I'm not.
Well, I'm glad you brought up this idea of if they've got X and Y and Z, and the diagnosis has essentially been put to rest by somebody, not us, we don't get that call. We have a parallel with the MH hotline that we don't know how many people are not calling us because they are competently handling an MH crisis and they never call us to consult us about it.
That's true. I mean, I get calls to evaluate patients who were diagnosed as children or people who had had an MH event as a child, was never confirmed. Now they want to be diagnosed because it's affecting their children's options for education or career, because you won't get into the military if the directive said that you have to have some of the directives said that you have to have a personal history, but a family history. And if the father so they'll want that person, military will want that person to have a diagnostic test, genetic tests, which are still in development.
Sure.
And as somebody said earlier today, a negative test does not outrule.
Right.
So the military will not accept that.
Right.
So those people will be sent to us for CHCT diagnostic testing. So
What would you like to talk about I haven't asked you questions about?
let me just add one thing about the military. I think I mentioned this earlier. If you were on active duty and you had an MH episode and you didn't know it when you came in, and now you are looked at, you might get a waiver. And those waivers, I just want to stress this, are entirely at the discretion of the service that you want to be in.
Depending on what critical service you could provide to them and where they could deploy you.
If they have spent $1 million training you to be able to detect whatever in the sky, they'll probably keep you. And they can profile a job description for you.
Sure.
We're not doing that for recruits. And the waiver system is currently under threat because there's a cutback in all services coming, we think. But that's something I want to be sure, because families are often told, oh, we have solved that, even by physicians. Oh, we've solved that. The military won't give you any problem. Your son is well qualified to get into the Naval Academy. Your dad was in it, you're in it. And that's just not true.
Right. What would you like to do over the next few years?
Read books. Go to the library.
Scientific, biographies, fiction? What do you enjoy?
No, I like my garden. I like traveling. I like going to Ireland. And I'm a history buff. And I've always been so.
When was the last time you were in Ireland?
A year ago. I'm going in October.
So
after the meeting in
Are you going to make it annual at this point then, you think?
Oh, I go on.
Oh, great.
Yeah,
Great.
my brothers.
Great.
My brother lives there, and my nieces and nephews and all that.
Great.
And lots of friends.
Great. Well, I want to express my personal and professional thanks to you. I appreciate the time and your willingness to sit down, and you prepared some questions, but you knew I was going to hit you with things that you weren't prepared for, and I thought you did great. So I want to say thank you on behalf of M-House, the board, the scientific community, and for all the patients and families you've served over this multi-decade career. My applause.
Thank you, Joe, and thank you for all the collaboration that we've had, all the things we've worked on together,
It's
and
been
that
my pleasure.
went so well because of that collaboration.